Welcome back. Today's whole lecture is all about muscles and movement. So we're going to dive into not really like specific muscles like origins and insertions. That's a little too specific, which is funny coming from IB. Something is actually too specific. We're going to be talking about joints, types of movement, rigor mortis, stuff like that. So let's get started. So in general, this is just sort of like your basic definitions, right? You have joints, which is like the area of pivot. So like your shoulder, your elbow, your knee, your hip, your fingers, right? Anywhere that like you can bend, that's a joint. Bones, I think you know what a bone is. Nerves and muscles, again, general definitions of what these are. You should absolutely know the difference between a tendon and a ligament. Tendons connect muscle to bone and ligaments connect bone to bone. But in general, these are your basic definitions. Now, the synovial joint is the only joint that we're going to get into this deeply, where you need to actually be able to draw, annotate, the whole 9 yards with. So let's start with our labels, right? So the muscle, synovial fluid, that's this blue stuff in between. This is helps with the shock. Joint capsule surrounds the entire joint itself. The tendon, right, muscle to bone. The bone, the cartilage, the ligament, right, bone to bone. And now let's talk about a little bit more of what this means. So the muscle provides force required for movement. The fluid, as I said, lubricates to reduce friction. This is also sort of like shock factor, right? Like you don't want your bones crushing against themselves. That is painful. This also provides nutrients to cells, right? Your bones have cells, your muscles have cells. Important to provide nutrients. The capsule is connective tissue that connects the bones and seals the joint, okay? Something has to keep that fluid inside, and this is what does that. The tendon, muscle to bone. The bone is the framework. The cartilage is smooth connective tissue to reduce friction and absorb shock. Again, this goes back to your synovial fluid as well. These two work in tandem. If you don't have cartilage, then your bones are like screeching against each other, like nails on a chalkboard. It's incredibly painful. This is arthritis, right? When your cartilage deteriorates and your bones start like creaking against each other. But cartilage has a major job in your body, and it's to absorb shock and reduce friction. This is why you can so smoothly like wiggle your fingers. And then ligament, bone to bone, which we've talked about. Again, you should draw this and be able to annotate it on a test. And when I say annotate, I mean label and explain what it does. So like both parts of this, the name and what's in all of these purple boxes. Now, the elbow is sort of like the joint that, it's not a synovial joint. Well, it is, but it's not like a ball and socket, which is like the ones that you typically think about. This is a hinge joint. But this is one that we're going to talk about a lot. We're going to focus on it when we do a dissection. So you should be able to both label and explain all of the pieces here. You should know the muscles involved, right? The bicep and the tricep, biceps in the front, triceps in the back. The bone, you have the humerus, then the ulna and the radius. Ulna is on the pinky side, radius is on the thumb side, and then the synovial joint itself. Going back to the slide before where you would be able to label and annotate it. So, focusing in on the elbow, bones, again, we've already defined that. You have the 3 bones there. So the humerus, which is like the top of your arm, acts as a lever so that you can move the muscles at the elbow, and then the radius and ulna act as levers for the 2 muscles that surround your humerus, the bicep and tricep. I think you're aware of the muscles and what they do, but in case you are not, these are the only real things that I'm going to ask you to memorize. in terms of muscles. So the point of origin and the point of insertion, I don't really need you to know that. But it's basically like where one area moves and one is connected to the bone. So the bicep muscle is responsible for flexing your arm, right? When you like flex, that's your bicep muscle contracting, right? You can feel that. If you like flex your arm right now, you can feel your bicep tight and your tricep is nice and loose underneath. If you do the opposite and extend your arm straight out, your tricep gets tight and your bicep is nice and loose. They are antagonistic muscles. Okay, they work against each other. So, different types of joints that we're going to kind of talk about. This is what I mentioned earlier. These are all synovial, but there's hinge, right? Elbow and knee, right? They don't have full range motion, but they can flex and extend. So you can go up and down, but you can't really like spin it around in a circle like you can with your shoulder and hip, which is a ball and socket joint. And that you can like do full circle movement. You can flex and extend, and then abduct and adduct, which is like move away from the body and bring into the body. In this case, the head of a bone sits in the cavity of another. So your humerus bone has a circular head that fits into your shoulder cavity. And same on your leg, your femur has a circular head that fits into your hip bone. I mentioned this, but now you have notes on it. Muscles are antagonistic. They work in pairs so that opposite movements can occur. Okay, so when one contracts, the other's relaxed. They perform the opposite action, right? So you can, when you flex your arm, you can flex and extend, right? Opposite actions, because those two muscles working are antagonistic. And then origin and insertion, just another time that I've given you this definition. At some point, you should write this down, though, just so that you have this, so that if you see this in a question, you know what these words are. Origin is where the muscle is attached to the bone, and insertion is where the muscle is attached to the moving bone. So you can see this here. The muscle is attached to the humerus. The humerus is not moving, but when you're flexing, your radius is moving and it's going up, right? So when your bicep contracts, it's pulling your radius towards the humerus. And of course, IB has to be weird and require you to memorize interesting things like an insect leg. So an insect leg has antagonistic muscles as well. The ones that we're going to focus on are the extensor tibia muscle and the flexor tibia muscle. So do the same thing. One extends the leg out and one flexes the leg in. It's like the same thing as your bicep and tricep, except on an insect leg. The exoskeleton itself, that's like the skin of an insect. It's made of chitin. That's how you say that word, chitin. And it's just like, it's like crunchy, you know? Have you ever like stepped on a bug and you hear the crunch? You're breaking the exoskeleton. Like an endoskeleton, right? Humans have an endoskeleton. Our skeleton is on the inside. The exoskeleton for insects is found on the outside. But like an endoskeleton, the exoskeleton does provide support and points for attachment for these muscles. Now, this is where it gets really a little bit specific, a little bit like really in depth. So muscle structure, and we're gonna go even deeper than this slide. So this is like your basics, and then we're gonna go deeper, and then we're gonna go like way too deep. So a bundle of muscle, this is from like big to small. So a fascicle is a bundle of muscle fiber. So like this whole thing is that. The fibers are these like individual, like almost look like Twizzler pull and peel pieces. They're just like multinucleated muscle cells, which means that they have multiple nucleuses in the cells themselves. And then myofibrils are within that. So one of these is this whole thing. So we're like zooming in, zooming in, zooming in. A myofibril is found within one of the fibers, and they're proteins, bundles of proteins. And then within that, there's a sarcomere, which is repeating contracting units, which sort of looks like this whole thing. We're going to have a close-up as we go. And then within that, there are myofilaments. Now, the prefix myo means muscle, so that's why you keep seeing it over and over again. Myofilaments. So it's just like the fibrils in the muscles, the filaments in the muscles. And the two that we're going to focus on for the myofilaments are actin and myosin, which are super tiny, and there's tons of them in this sarcomere, and we'll talk about that more in the next slide or two. I guess the next slide. So actin and myosin. Actin is thin, so it's like the tiny one, whereas myosin is thick. So myosin, when you see a picture, will be the bigger of the two. Actin has two proteins on it, troponin and tropomyosin. You will need to understand the two and how they work together. And actin has a binding site for myosin heads, which are these like golf club looking things. So these myosin heads actually connect to actin. And we're going to talk about how that happens in a minute. But this is how these two myofilaments are responsible for muscle contraction. So yeah, the only thing I'm missing on this is that myosin heads contain actin binding sites, right? Because myosin binds to actin, right? And there's ATP involved, right? You need energy to be able to move muscles. Before we get into how muscles actually contract, which is the sliding filament theory, let's talk a little bit more about the muscle cell. So sarcolemma is the cell membrane of a muscle fiber, right? We've already talked about a muscle fiber, but now we're like zooming in on that and focusing on all of this. And I know this is so much vocab. I know it is. You need to be familiar with it. I'm never going to ask you to like straight up give me these definitions, but you would have to be able to confidently use them in explaining muscle cell structure, like just explaining like how it's set up, or in a multiple choice question, these words will be used and you will have to understand what they mean. The good thing about this is you already know what some of these words mean. So sarcolemma is the membrane, but sarcoplasm, you've heard plasm before in terms of cell. It's the cytoplasm in a muscle cell, in a muscle fiber. So sarcoplasm, cytoplasm, sarcoplasmic reticulum, endoplasmic reticulum, right? It's like you've heard these. It's just that we're using a slightly different prefix. A T-tubule is the folding in of the sarcolemma, which allows the impulse, the nerve impulse, to travel along the muscle fiber, which tells the muscle like, hey, you need to contract now. Mr. Brain is telling us that we need to move, right? Your brain is responsible for sending nerves out. The sarcomere is what we talked about already, the contraction unit. The sarcomere is made up of myosin and actin, and then mitochondria, powerhouse of the cell, abundant for ATP production, right? You hopefully knew that part. Now, diving further into sarcomeres and muscle contraction, I will make you draw this. If you don't draw it in your notes, you will be drawing it in class. I guarantee it. This is a complicated piece, and I'm going to make sure that you guys understand all the parts. A bands, I bands, H zones, Z lines, all of that is a sarcomere. So from Z line to Z line is one sarcomere. You have the actin, which is the green, and then you have the myosin, which is the purple. So from myosin to myosin, right, you see that like from the heads to the heads, that's the A band. Actin to the other actin, not including the myosin, but is including the Z line, that's the I band. And then the H zone is strictly just the myosin, but this H zone disappears when the muscle contracts. We'll talk about that more on the next bit. So, okay, well, I just I just explained all this on the picture. So you can pause and write this down if you want. But again, actin is thin, myosin is thick, Z-line is the end of a sarcomere, I-band is just actin, A-band is the whole myosin, and H-zone is just myosin. The A-band is the only one that has both actin and myosin. Now, the sliding filament model, sliding filament theory, you're going to hear both. Actin slides over myosin. So again, actin is this lighter one, the blue in this picture. Myosin is the red. Actin is going to slide over the myosin. You can see it here. They're sliding over each other. And by doing that, the muscle is contracting. Okay? So action potential is your nerve, right? Your nerve impulse. So your AP, action potential, reaches the terminal end of your motor neuron. Basically, the neuron that attaches to this muscle, the impulse gets sent to that neuron, and that neuron releases acetylcholine, but really, it's starting the whole process. It's saying, all right, buddy, let's go. We got to contract. We got to run away. There's like a bear chasing us. I don't know why you would be in that situation, but something is happening and your muscles need to move. Okay, so the action potential reaches the muscle, releases acetylcholine. Acetylcholine binds to the sarcolemma, right, the outside of the fiber, and it starts an action potential. An action potential is, again, that impulse is traveling. That same impulse that was originally being sent, it's still being moved, okay? It's traveling through the muscle cell. It's carried to the sarcolemma, down into the T-tubule, and it triggers calcium. Ding, 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 key point, highlight, underline. Triggers calcium from the sarcoplasmic reticulum. Calcium is released. Once calcium is released, this is where it gets interesting. Calcium gets released, and it binds to troponin, which is on the actin, right? So calcium gets released, binds to troponin, and what that does once calcium binds to it is it moves tropomyosin. So the calcium binding causes a chain reaction and tropomyosin moves. Now those binding sites are open. So myosin is like, hey buddy, I haven't seen you in a while because you've been hidden away, because it's usually covering the tropomyosin is usually covering the binding sites. But now it's open and myosin wants to hang out. So myosin forms a cross bridge with actin. Basically, these heads of myosin decide to hang out and they connect. You need energy for this. So energy causes the myosin head to move, and it moves against the actin. And it's basically, well, it's called a power stroke, but it's basically like reaching out, grabbing onto actin, and yanking it forward. Like right now, stick your hands out in front of you, grab on, and then pull your hands back. It's doing that motion. It's grabbing onto something and pulling it towards you, right? So that's what myosin heads are doing. They're attaching to the actin and then pulling it in. Once they are pulled all the way in with that energy, myosin heads detach and then reattach to actin further down and continue pulling the actin inward. This actin slides over myosin. This power stroke happens again, and it happens again and again until the muscle is contracted totally. And that is how your muscles contract. So just in case we missed all that, right? It needs energy. Once energy is used for the head to attach to the actin, it then comes back down, right? It pulls itself, the power stroke happens, and it pulls actin and starts to contract a little bit. And then you need to start all over. Another ATP molecule needs to come in to do this. You cannot do this without energy. Energy is absolutely vital. All right. This is getting to the end here. So this is rigor mortis. I think there's like one slide after this or a couple slides after this. Rigor mortis is what happens to your body after you die. You can actually measure out how long it's been since a body has stop being alive, based on how stiff the muscles are. So what happens is you're not making energy anymore. So there's no ATP left in your body to take the myosin heads off of actin. So the myosin just keeps your muscles in constant contraction, and it's doing this process. It's like still on like the power stroke, but it never lets go at the end. And so your muscles are just contracted. Eventually, it's just fatigued, and it will eventually let go, and it will relax. It will stop contracting, but that takes hours. So you can actually measure the time of death using rigor mortis. All right, last slide. Using fluorescence. This is how we figured this out, is we like dyed muscle fibers with a fluorescence, and then we were able to actually see. So injected with the protein, this bioluminescence coincides with the release of calcium. So we measured that. And this is actually like this, well, not this experiment, but this did show that calcium is like the main contributor to making this happen. But another experiment actually attached fluorescent dye to myosin, and that's how we know that the power stroke happens and how myosin sort of like creeps along, like attaches, pulls, let's go, attaches, pulls, let's go, is because we fluorescently dyed them and then watched it happen. And we could see it because it was bright green, and it's very obvious what's happening when things are bright neon green. Okay, that's everything for this one. Great job. I will see you in class where we will do some dissections.